Hello students, this is your instructor Abid Jan and I will proceed with NEB Cutter from the course of Bioinformatics. NEB Cutter is actually a program to cleave DNA with restriction enzyme. The two sides of a DNA sequence. Deoxyribonucleic acid, DNA, the stuff that makes up our gene, is a large macromolecule. It is a long chain-like molecule twisted into a double helix. In 1953, Watson and Crick elucidated the famous double helical structure of the DNA molecule. These days, everybody has a mental picture of this famous spiral staircase molecule. But what made this discovery so important was not the helical shape, but the discovery that the DNA molecule consists of two complementary strands. And this figure displays the two complementary strands of a complete DNA molecule. This is a complementary strand, double-stranded DNA complementary strand. This is one strand running from 5' to 3' direction and other strand running from 5' to 3' direction. This is anti-parallel double-stranded DNA. Now, what is complementarity? By complementarity, we mean that a thymine on one strand is always facing an adenine and vice versa, and guanine is always facing a cytosine. These couples A to T and G to C have a strict one-to-one reciprocal relationship. When you know the sequence of nucleotides along one strand, you can automatically deduce the sequence on the other one. This amazing property of complementarity and not the stylish helical structure is the key that explains everything about DNA sequences. For instance, when living organisms reproduce, each of their genes must be duplicated. In order to do this, nature does not go about it the way a photocopier would, by making an exact copy. Rather, nature separates the DNA strands and makes two complementary ones. While reading the nucleotides from the 5' end towards the 3' end, you may decide to write down the bottom or the top sequence. Remember, both strands are not equally valid sequences. Thus, at each location, a DNA molecule corresponds to two totally different sequences. Fortunately, most database mining programs such as BLAST know about this property and take both strands into account when reporting their results. But some programs do not bother and only analyze the sequence you gave them. So in cases where both strands matter, always make sure that a complete analysis has been performed. Palindromes in DNA sequences. Newcomers to DNA sequence analysis are usually confused by the notion of reverse complementary sequences. However, in due time you will be able to recognize the relationship between both strands of a double-stranded DNA molecule. One property of DNA complementarity is the fact that regions of DNA may correspond to sequences that are identical when read from the two complementary strands, and this figure helps illustrate this trick. 5 to 3 this way and 5 to 3 this way. So 5 to 3, this is actually TGA, TCA. Now 5 to 3 this way, TGA, TCA, and at the same time the reciprocal relationship is satisfying. A to T, G to C, A to T, A to T, G to C, A to T. So the A to T and G to C relationship is satisfying, and from 5 prime to 3 prime on one strand going from right to left, for example the above strand, and 5 prime to 3 prime, the below strand going from left to right, the read is the same. So such reads and such patterns are called palindromes, after the term for a phrase that reads the same in both directions. For example, MADAM from left to right, and now MADAM from right to left, the read is the same. Such sequences are called palindromes. Biological roles of palindromic sequences. Palindromic sequences are not merely a curiosity, they play important biological roles. For example, most DNA cutting enzymes have palindromic recognition sequences. Other palindromic sequences serve as binding sites where regulatory proteins stick so they can turn genes on and off. So this is a regulatory role. Palindromic sequences also have a strong influence on the three-dimensional structure of DNA molecules, and therefore looking for exact or approximate palindromes in DNA sequence is a classical bioinformatics exercise. NEB Cutter. NEB Cutter is a program available via a web server of this URL that will accept an input DNA sequence and produce a comprehensive report of the restriction enzymes that will cleave the DNA sequence, the given DNA sequence. NEB Cutter will take a DNA sequence and find the large non-overlapping open reading frames using the E. coli genetic code and the sites for all type 2 commercially available type 3 restriction enzyme that cut the sequence just once. By default, only enzymes available from NEB are used, but other sets may be chosen, and You just need to enter your sequence and submit. Further options will appear with the output, and the maximum size of the input file is 1 megabyte, and the maximum sequence length is 300 kilobases. NEB Cutter produces a variety of outputs including restriction enzyme maps, theoretical digests, and links into the restriction enzyme database, REBase, REBase, restriction enzyme database, and importantly, its table of recognition sites is updated daily from REBase, and it marks all sites that are potentially affected by DNA methylation. Many options exist to choose the enzymes used for digestion, including all non-specificities, subsets of those that are commercially available, or sets of enzymes that produce compatible termini. Now we need to point our browser to two biological databases. One is Ensemble, and this is actually a DNA sequence database, and the other one is NEB Cutter. So this is the home page of the NEB Cutter version 2.0. As you can see, NEB Cutter accepts, this is the input box, and NEB Cutter accepts an input sequence in plain or FASTA format, which can be pasted in, picked up from a local file, or retrieved from Ensemble Genome Browser or NCBI as a gene bank file through its accession number. And as you can see from here that various options are available to select the size of open reading frames to be displayed and the set of restriction enzymes to be used. So a different set we can choose, and we can adjust the size of the open reading frame. For example, now we need a gene sequence, so I will click on the human, and in the human, suppose I will go for a gene symbol, for example LIPH. This is a human gene. Now this gene has 5 transcripts and 4 are coding. So suppose we are investigating this transcript, the transcript at the top in the table. So click on the transcript ID. Now click on the cDNA. We will get access this way to the cDNA of the LIPH, and actually we need the sequence as an input for the NEB cutter. Now click on the axons. So this is, and click on the CCDS of the LIPH. So this is the cDNA of the LIPH. Copy it and paste it here and submit it. Now this figure is actually the linear display of a digest of molecule LIPH. Now suppose we change and adjust the reading frame to 30. Or we try to reduce it further to 15. So this way we can adjust the length of the open reading frame. In this initial display there are many options to go further. For example, custom digestion with enzymes of your choice and various displays of the target. It is also possible from the linear display. This is the linear display. To zoom in a selected region, for example, suppose we click here and then zoom in. So I can get back, unzoom. Now this is the first initial linear display. So actually this allow a particular region, this zooming in, a particular region to be selected, and those enzymes that cut immediately adjacent to the region to be displayed. So this is useful in order to find enzymes able to excise the desired region. We can find out those enzymes through which we can excise that fragment from the plasmid or from the entire gene. And on any main display, moving the mouse or the cursor over restriction enzyme name, for example these are all different restriction enzymes, it will show a recognition sequence. For example, for this ACI1, the recognition sequence is CCGC. And also the precise base number at which it cleaves. So it cleaves at 851 nucleotide. And if more than one site is present for the enzyme, then all other sites are underlined. For example, we cannot see any enzyme underlined because there is only single site for each enzyme. In case if there are more than one site for the same enzyme, then the enzyme is underlined. Now clicking on the enzyme, for example, I click on ACI1. So clicking on enzyme will produce a page with a summary of information about the enzyme, including its methylation. This is the methylation detail of the enzyme and the kind of ends produced. So this is producing overhang, and it also incorporates everything that is recorded in the rebase enzyme page about the methylation sensitivity of any enzyme. Also we can see the information about digestion condition. So on every page, hot links are provided that lead back to the main page, provide help in interpreting the display, or allow users to send comments back to the authors. And the interface has been designed to be as user friendly as possible and to provide easy access to the main functions useful to researchers attempting to cleave their DNA. So it was all about the NEB cutter, and this is the bibliography. Keep calm and stay curious. All the best.